Good morning, everybody. Um, this is going to be, I don't know, maybe I'll try making it as interactive as possible, even through the Zoom, because sadly I can't see anybody in my class, and that that kind of, I mean, we'll see. I hope I'm not just talking to the screen, and I'm talking to students across the screen. Okay, so I understand that. You have been briefed substantially about development per se, right? So can anybody tell me, can anybody brief me about what all has been covered in the class as to, I know Raj has mentioned that he's touched upon a lot of things, um, described concepts and spoken about many things in development in general. But I just wanted to hear out from some of you, or maybe one of you, about what has been covered in the past classes. That will help me pick this lecture up from where you've stopped. Can people hear me?
Yes.
Yeah. So
Hi, last class was about mathematical.
who's this? Can you introduce me yourself?
Sorry, I'm Anindita, and I'm part of the integrated PhD batch 2020.
Okay.
And Raj has talked about the gradient establishment, the morphogen gradient establishment, and slightly touched upon the genetics. And we had our last class on basic mathematical modeling, like the basis of it.
Okay. 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 So you have, so anything about mouse development, axis specification, anything on those?
Yeah.
Okay, so you have have an idea of how a single sheet of epithelial cells is then transformed into, kind of gone over the development of the early developmental stages of, say, mouse. I don't know. So he has touched upon that as well.
So we've just not specifically mouse, but we've discussed the general process. We
Okay.
haven't discussed specific models. He's given us examples how it's comparative in Xenopus, chick in humans, and yeah.
Okay.
We
Okay.
haven't specifically gone in details of one particular model.
Okay. Okay. So okay, fine. So probably this is then the first series of lecture. We will try to see how a fly develops. And this has been thanks Anandita, thank you so much. This has been a very important model system, not just a useful, but a fundamental model system for um, uncovering the foundations of development. And the usefulness of this system was only later on realized. I mean, earlier on, when people were actually understanding or going over embryology, looking into development, Drosophila didn't come across as one of the most useful model systems. Precisely because it's very small, the embryo always needed, you have to visualize the embryo under the microscope, and not much can you see that happens in great detail, as opposed to Xenopus, chick, which were the more favorite model systems. So embryology experiments, transplantation were much more easier in those systems. A, the size, B, you can technically could do transplantation, and C, the visualization was also much easier. But Drosophila was not a very favorite system. it was easier to work with, it was easier to grow, cheap, and all of that. But for embryologists, it wasn't a model system that they would be intending on exploring till the onset or advent of molecular biology. This is where I feel integration of two different fields have made two fundamental discoveries. So soon the advent of molecular biology, genetics could be possible, and you could like isolation of RNA, DNA, and understanding the development of these molecular tools became available. Drosophila suddenly picked up as one of the most important model systems for understanding development, because genetics of the system became this was the forte of this model system. You could employ genetics. Manipulating this system became like the advent of p elements, the utilization of and being able to isolate DNA and doing cDNAs and trying to see where the genes localize, identifying enhancers. All of these became possible with molecular biology, and coupled to genetics that this system offered, fundamental points got laid out through this model system, and since then this has been a very useful system, not just now for development, but in general understanding many different concepts, be it cancer, be it stem cells, be it anything. Okay, so I would suggest that if there are questions, please feel free to stop, because I don't see anybody. I see complete silence across. So please make sure that if you have a doubt or if you feel there is a point that you would like to make, please feel free to intervene. Okay, so this is, and I work on Drosophila. So as personally, our lab works on Drosophila blood development. And I think that this is, I mean, if one could understand how one can make a fly, then I guess then you can start thinking of making other or building other complex systems, because even if it's a fly, it's quite difficult to, and when you see how it develops, it's not that simple. Okay, so this slide shows the life cycle of Drosophila, and you have a male and a female fly here, and their life cycle starts with mating, and the embryo is laid, and you can see how small the embryo is. This embryo undergoes from the embryo, which is a 24-hour time point. The embryonic development is 24 hours. From the embryo, the first instar larvae hatches, and that in 3 days grows exponentially to a third instar larvae, which then enters a pupa, stages of pupal development, and then transitioning into an adult from here on. So this has stages of transitions, and this is a holometabolous insect animal, because meaning that the ultimate adult doesn't resemble any of the stages that it develops from. The system is actually, so the usefulness of the system is that not only they just eat and grow, but it's quite a cheap system to work with. So maintaining, I mean, the reason it was quite a popular model system, and it started with Morgan's lab, where they were employing genetics, and I think one thing that the culture of Drosophila, the culture that Drosophila has developed, is that it's a very simple system to work with, even for undergraduates or school kids. And this culture was developed since the system was being utilized. So in Morgan's lab, all the undergraduates, Calvin Bridges, Alfred Stuttervant, they were working on this model system, and they would score hundreds of flies for thousands and thousands of flies for genetic variations that they would find, and then try to utilize genetics, recombination mapping to see where the mutants would lie. And it is not very intense in the context of physical labor. I mean, it just requires students sitting under the microscope and just scoring what are the different variants that they find in just the population. So it is visually scoring the flies. And this led to a lot of identification of those genes, like the white-eyed phenotype or the yellow body phenotype. These were all discovered by undergraduates. And I think this culture is very good because it instills, that back then, it instilled the idea of asking a question and then answering it through your own means. So intellectually also it stimulated kids from a very young age. Okay, so moving here, so we start with how does the egg develop and where does it come from? So Drosophila fertilization is unlike the fertilization events that you might have been introduced to, because the sperm entry activates the egg in other systems. But unlike the other systems or the mammalian or vertebrate systems, the egg here, as it is already prior to the sperm entry, is kind of partially activated. This is because as the egg as the egg travels through the ducts, the squishing action of the ducts actually lead to the activation of the egg and the oviduct, I mean. And as the egg squeezes itself through the oviduct, this actually leads to the activation of calcium channels, and this kind of pre-activates the egg. Following which the sperm entry happens, and then the complete fertilization is achieved. So as the calcium channels open, the eggs start, the nucleus, which is halted in its division, resumes its meiotic division. And now all the RNA that has been loaded into the egg also start to be translated. Not entirely, not the entire mRNA is translated, but translation is activated now. So the stored mRNAs that are there through the maternal components are being translated. And now since the sperm entry happens, then the egg becomes completely active. Now what happens here, so how does the sperm, through what does the sperm entry happen? This is also directed. So the axis of the point of entry of the sperm is already pre-decided. It's not that, unlike the mammalian context where the sperm entry happens, is not decided. But here in the Drosophila egg, so if you see the two tentacles that I'm pointing out here, that is called the micropyle. And it is through this, the micropyle is when the sperm entry happens. So it's already decided that this is going to be the axis of fertilization, and this is the axis of entry. And the micropyle does two things. A, it directs where the sperm should enter from and fertilize the egg. And second, it also restricts polyspermy, because it allows, this is very narrow, so it only allows one sperm to enter at a time. So so polyspermy is prevented like that. And unlike other fertilization events in other models, here the entire nucleus and the sperm tail enter. The sperm tail actually are released into the egg. So it is the complete, and the entire sperm gets incorporated into the oocyte cytoplasm. So it is not that just the nucleus enters, it's just the genetic material that is incorporated. It is the entire sperm, the tail, the nucleus, entire thing gets incorporated into the oocyte cytoplasm. And until the entire sperm enters into the egg, the sperm membrane, the membrane of the sperm cell doesn't break down. So this is quite a stark difference from the other systems. Now, the other thing is that since the egg is partially activated, with the entry of the sperm, now it becomes completely activated, because this will result in the this will result in the activation of complete activation of translation, and the early maternal components that have been laid and the translation that was initiated will now begin full swing post fertilization. Is this okay? Is everybody following?
Yes.
Okay, great.
I have a question.
Yeah, sure.
This is Nivedita.
Hi, Nivedita.
Yeah. So I wanted to ask that you talked about eggs being partially and then completely activated. So are the molecular changes differentiating between the partially and completely activated states only in the translation of the mRNA molecules?
So, yeah, because now it's a haploid, right? So the oocyte will be haploid, correct? Now with the entry of the sperm, it's going to be a diploid, right? So now the meiosis is going to be completed. What do you call? Not just the translation, but the genetic. It's not going to be transcriptionally active yet, but the entry of the sperm is going to be driving a robust translation of all the maternal components, plus the genetic material is now going to get ready for transcription to start off, which will not happen immediately, but will happen in some time.
Yes.
Yeah?
Yeah, got it.
Okay. So now what happens next is now it's an embryo. So it has, now this is what it'll look like. So the stage 1 that you see is post fertilization. Now you have that nucleus and this huge cytoplasm. So this is what it ends up. Now, while translation was going on minimally, translation of the maternal RNAs will now kick in more robustly. So now the point is, this is quite different again in Drosophila as opposed to the development that you might have seen in vertebrates. So in other systems, the egg divisions are complete, meaning there is cellular divisions, correct? Complete with the cell membrane dividing the different blastomeres, correct? Now in Drosophila, it is not the same. The first few divisions in Drosophila are just superficial, meaning that the nuclei divide. The nuclei undergo SM, synthesis and mitosis, But there is no cytokinesis that happens, meaning there is no cell membrane based divisions. So ultimately it is one egg it's a multi-nucleated egg, right? All the way until stage 9, 10, it is a multi-nucleated egg. So it's called a syncytium. And it is only later on, from stage 13 onwards, is when the actual cellular divisions are seen. So till all the way from stage 1 to stage 12, or even, 12 is probably when the cellular membranes start to pinch in and separate the individual cells. But till then, this is a single cell carrying multiple nuclei, something like the muscles are. So it's a syncytium. Maybe one question I would pose here to you guys that you can think upon or ponder upon as to why is this the case in Drosophila, and why is this not the case in mouse? Why does the mouse have cellular divisions right from the beginning? And why is the Drosophila dividing like this? I mean, utilizes a syncytium-based module. So it divides, the nuclei divide and give rise to multiple nuclei, but it's just one single cell till some point. And only later on, the cellular membranes develop and that then divided into multiple cells. So something that you guys can ponder upon, and maybe on Wednesday, we can touch upon some thoughts that you might have gathered. Okay, so now, as you can see here, I've written that the early divisions are very quick. So within 8 minutes, one division, nuclear division happens. And and within 3 to 4 hours, all of this is achieved. So if you were to actually take a Drosophila embryo, it's quite stunning because you can see it happen in front of you. You take Drosophila embryo at different time points at every 8 minutes, and you will see, as you see, or even if you live image, or as you even fix embryos of different time points, you will see that within 8 minutes, there is a dramatic difference in the number of nuclei. In the beginning, they're very synchronized divisions, so it's visually very stunning. I will probably try and pull out some videos here for you, and maybe I'll share it as a Zoom link, share it so that you can see it. They're very visually stunning because it looks like a synchronized dance of the nuclei. And later on, it gets asynchronous from cycle 14 onwards, when more different kinds of events start off. The divisions are no longer synchronized. So they're fast and very synchronized, and that leads to a syncytium, generating a cell, which is syncytium. And later on, going into an asynchronous cycle, which will then lead to the following events, which I'll come to in a minute. Now, one of the key things that need to be, that I will probably focus here are, so as these early nuclear divisions are happening, which are very rapid, around this stage 9 or around the stage 9 or 8 or 9, a few nuclei get specified that'll become the pole cell. So it happens very early, around stage 8 or 9. There are a few nuclei at the posterior end of the embryo. They gradually move towards the posterior pole plasm, as you can see more clearly in picture 12, stage 12. So they get specified, start getting specified around 8 or 9. How they get specified, what are the events that are leading to specification is still something that not very clearly understood, but I mean, to a large extent understood that there's probably something in the posterior pole plasm that leads to the specification of some nuclei at these stages. So it's the pole plasm here. Pole plasm meaning at the posterior end, the cytoplasmic determinants are not the same as the anterior end. And the cytoplasmic determinants at the posterior end, however, sensitize a few nuclei to migrate towards them. So sensitized a few nuclei that will form the primordial germ cells. So they're called pole cells, and which will give rise to the future gonads. So they're called the primordial germ cells. And they then start migrating, unlike all the other nuclei which will migrate to the periphery. These pole cells will migrate to the posterior end now, which carries the pole plasm. And from here on, these are the cells which will be now giving rise to the gonads. So this is the first specification that happens of not cells, but of the nuclei. And later on, as the cellularization happens, they will give rise to the primordial germ cells. So the nuclei, even though they will be all similar, from stage 8 or 9, one can start seeing that specification is beginning to get initiated. And the first specification that one sees is the specification of the pole cells. And their migration, which is now not going to be towards any random direction, particularly towards the posterior pole plasm, is indicative of that now things are changing within the embryo. It's not the same between the anterior and the posterior end. So do I make sense here? Okay, great.
Yes.
Okay, so now as you can see the syncytium with all the nuclei in between. Now the posterior pole cells are going to migrate. Now what the other nuclei are going to do are move towards the periphery. As they move towards the periphery, they're also dividing, right? So, but now the divisions are not going to be that synchronized. They're going to be also time-taking. They're going to be longer divisions. They might last from, I forget the times, but unlike 8 minutes, sometimes it can be 75 minutes to, I'm forgetting the times. Maybe as from 75 to as long as 2 to 3 hours, 2 hours also. So it can be longer cell divisions. From stage 30, more later. But you can see that the nuclei are also increasing in number, right? So the divisions start, the timings slow down, but the divisions continue. But the nuclei also are moving towards the periphery. And from here, from stage 12 onwards, what is going to happen is this process called cellularization. So until now, nothing is divided as single cells. You have the yolk, and you have the cells surrounding now. But from 12, 13, and complete 14 stages, there's going to be this process which will lead to the formation of individual cells. So now what is going to happen is that as the nuclei move towards the surface of the egg, both the microtubules and the microfilament-based functions become very critical here. Because what they're going to do is drag the cell membrane of the egg and lead to formation of and separate the cells from the yolk. So this is going to lead to the formation of individual cells now. And at this point of time, the actin filaments become very critical for this process. You can read about experiments that have led to this finding, because those are very, I mean, they've just used actin filament, actin polymerization blockers, and they see that the development of the egg stops. Now, how does this happen? The microtubules pull, and the furrows that are generated that divide the individual cells. So there's a complex dynamics between actin, microtubules and microfilaments that lead to the formation of individual cells. And maybe I will just, after I finish the slides for today, maybe I will show you some videos where these divisions are happening, and they look very nice. Okay, so this will lead to the formation of so earlier it was a syncytial blastoderm. Post this, stage 13 onwards. So now from 13 to 14 is where it is completely cellularized. So individual cells have formed. This is what it was at stage 13, and this is what it is at stage 14. And there is an apical basal polarity that gets established just because the nuclei are more apically based and apically placed than basally. And this is called the cellular blastoderm. And this transition is called cellularization. Now, following the cellularization, now you can see that the embryo has been, the egg was fertilized. The egg started dividing its nuclei, and it became a multi-nucleated single cell. Then it made a few cells from the posterior end move towards the pole, posterior pole. The first specification is the first event that leads to specification is the event that leads to the formation of pole cells. And thereafter, now the pole, before that, there is no specification that happens. So the formation of the pole cells separates what is going to be the future gonad. And then the events that lead to the formation of the cellular blastoderm are initiated. And now you have cells surrounding the yolk, all surrounding the yolk that have been set up. Now this is where the next stage of development starts off, which is gastrulation. Now, I mean, I think Woodward has a famous saying that if you don't gastrulate, you would, I'm forgetting the saying, but there's a very nice saying that he's made, that one of the most important things in development is you have to gastrulate. So, and if you don't gastrulate, then essentially you cannot develop. So, can anybody tell me what is gastrulation and what does it lead to?
Gastrulation is the process by which the single-layered blastula becomes a double-layered gastrula, and the three germ layers, the ectoderm, endoderm, mesoderm, are established.
Great. Who's this? Can you introduce me? Can you introduce yourself to me?
Yeah, I'm Shubha, and I'm a integrated PhD student of the GS 2020.
Okay, great. Thanks, Shubha. Thank you so much. So yeah, Shubha, well said. So gastrulation, so this is clearly a single layered group of cells. Now it becomes double layered, absolutely correct. And as it becomes double layered, it separates the three germ layers, ectoderm, endoderm, and the mesoderm. Now, this is what happens in the way gastrulation happens in Drosophila is what this picture shows. So now all the colors here are colors that imply um, different different germ layers, but they're not being defined as of now. It's in the process of gastrulation. They will get specified. Now, the first event that happens in gastrulation in a gastrulating Drosophila embryo is the formation of the mesoderm. Now, this is anterior, posterior, dorsal, and ventral. So, at the ventral side of the embryo, you have a group of 1,000 cells or so, which start moving inwards. So you can see that they're moving inwards. So this is called the ventral furrow. And as they move inward, they segregate these group of cells from the rest. So this forms the mesoderm. And surrounding the mesoderm are the ectoderm cells. So which will give rise to the neural ectoderm or the internal ectoderm. And the now as these cells move in, now what happens is that the gastrulate these ventral cells, as they move in, they're going to you can see here they're moving. They will move more towards the posterior end and curve up. And at this point of time, they carry the germ cells as this movement is happening. So there's a lot of movement that will happen now. They will carry the germ cells and place it inside, so which will be the site where the future gonad is going to develop. And you can see that they're going to go like this. So this is called germ band extension. So this ventral furrow will segregate a few group of cells that will give rise to the mesoderm, but also this thing is called a germ band, and because it carries the primordial germ cells, and this germ band is going to give rise to structures of the thorax and the abdomen. So these group of cells, unlike the others, ahead and behind the germ band, which will give rise to the head or the tail, this particular event of the germ band will give rise to the thorax and the abdomen. The germ band extends posteriorly, curves upwards, because it can't go straight. The embryo is confined within its shell. So it extends, places the primordial germ cells, and the extension places the primordial germ cells inside. Formation of the hindgut, midgut happens, the gut gets specified, and then following this, the germ band will retract. We'll come to that in a bit. So looking at how the ventral furrow is. So you can see at the ventral end, there is this furrow. So the cells start moving inwards. So they gradually curve up in, and there's this process called convergent extension. We'll come to that maybe later. They start moving in. This forms the ventral furrow. And at the same time, as the ventral furrow gets formed, at the anterior end, which is going to give rise to the future head and the head structures, the antennal structures, there is this other indentation that gets specified, starts initiating, is the procephalic furrow. So the head, this kind of bends a little forward and retracts. So this leads to the formation of cells getting in to formation and leading to the formation of the cephalic furrow. So two things happen, the ventral furrow and then leading to the formation of the cephalic furrow. So you have segregation of the head thorax, abdomen, and also part of the head here also gets specified from the germ band. But, and the movement and the formation of the posterior pole cells, separation of the posterior end, which is the pole cells. So yeah, now from the ventral end, you can see here, this is the cephalic furrow, and this is the ventral furrow. And once this has happened, then you have more different events, morphogenetic events that will lead to the formation of other structures. Now, the anterior midgut is specified from this head region, but the posterior midgut, hindgut is specified from the ventral furrow. So the gut has structures that come from. So if the gut is taken as anterior midgut, mid-midgut, posterior midgut, and hindgut, you have the gut which is coming from different sections of gastrulating embryo. So the anterior gut will come from, midgut will come from this head invagination, when process of head invagination is happening, whereas the posterior midgut will come from the cells which are moving the pole cells into. Okay, so this is the germ band, the cells of the germ band, which will extend all the way to closer to the head. The procephalon is going to give rise to all the head structures. And you can see at this point of time, what is going to be the posterior part of the embryo is just next to the head, because they have all moved, they've all stretched themselves out, and given the cuticle, the eggshell prevents it from moving all the way this side. So that is why perhaps they curl up. And then these different indentations, you can start seeing segmentation happening. So, which means that what I mean by segmentation is that once this germ band has completely extended, the events that separate the germ band into repeating units gets initiated. Now, so you will have the repeating units, they look all the same, but they're not going to, so the germ band is going to get divided into different units. They might look the same, but they're not going to give rise to the same structures. The first few units are going to give rise to the head, the thorax, the abdomen, different units of the abdomen. And following this, so you can see what the germ band is actually doing. It's segregating now the group of cells which have been formed into different structures of the animal. Now, following this germ band, once the segmentation has happened, in late events of gastrulation, this germ band is going to move backwards. So it's like stretching a rubber band all the way to curve up onto the top and then releasing it. So as it releases, it goes back. So now the segmentation is complete, the structures have formed, and it's getting closer to something which will look like a first instar larvae. And it is at this point of time, the germ band movement retracts, and this is still, there's this yolk which is still in the animal, and it's still an open, the embryo hasn't closed. So meaning these segments are separated by this layer of yolk, which is called amniocerosa. And, but anyways, organogenesis, more segmentation, and development, a specification of specific structures that are going to be in the larval stages of development is happening at this point of time. So a lot of organogenesis is happening, and this stage is very critical. And after this, where is the closure? Sorry. Okay. what this picture doesn't show here is the closure of the two ends. So it looks like this. I'll show a video in a bit. So the two ends will meet and close, which is called the dorsal closure. So this is dorsal and ventral. Now the two ends of the embryo are going to meet, and this amnioserosa, which is the leftover cells of the yolk, are going to be not deleted, but they're going to undergo apoptosis, and the cells will move closer together, close the embryo, and which will be called as the dorsal closure. Okay, so before I move to the video, I just want to, is this, I mean, it'll look much more clear once we see how it happens through some videos. Now, as you can see that the embryo is going to undergo a lot of changes post stage 14, right? So now, what is leading to all of these changes? How is the embryo doing it? Is one question that we need to ask. Has the genetic material of the embryo started to kick in? Is it active? Is it transcribing? Is it still dependent on maternal components? Is what we need to ask. So earlier on, from the early divisions are actually maternally driven. So all the way to stage 11 to 12, it is mostly maternally driven. So while the maternal components were initially being translated, so it's a lot of RNA and protein that are laid by the mom into the developing oocyte. And as the oocyte might, moves through the oviduct, the squishing of the oviduct, and that activates the egg, leads to a little bit of translation initiation of the maternal components, and which is then taken over full swing with the entry of the sperm. Now, it is heavily maternally driven still after that. So all the early stages of division are being still driven by maternal proteins and translation of maternal RNA. So if any translation questions need to be posed, the early stages of embryogenesis across systems is perhaps the best, because transcription actually doesn't even have a role to play at these early time points. It is mainly translation that drives cellular processes. And how translation does this is one active area of research. Now, following the translation that happens, the zygotic genome kicks in. So once the zygotic genome is kind of needs to be activated as well. So now the maternal components lead to now transition into the zygotic components. So meaning now the zygotic genome is slowly going to start transcribing. It is slowly going to start transcribing and translating. So now the embryo is going to take over and be the driver, driving the subsequent processes from here on. So the zygotic, it's not that the zygotic genome will start transcribing, boom, and everything starts. No, it is a very gradual process. So it begins around stage 11, around cycle 11. And following this, it starts to increase. Now, as the zygotic genome gets transcriptionally more active, what is seen is that the nuclear divisions slow down. The cellularization happened. So the cellularization event is seen post the activation of the zygotic genome. And now, since the zygotic genome is transcribing, you start seeing zygotic RNA being expressed. Now, what is laid by the mom is very specific. Now you can see, one can start seeing uRNA being present. This is now, but it's like a very young zygotic genome which is starting. This is called transition. It is not yet they're still transitioning. So this is the mid-blastular transition. And now, even here at this point of time, as the zygotic genome gets activated, this mid-blastular transition initiates more robust activation of the zygotic genome. What is also seen is that the maternal components are going to slowly start getting degraded. So there is going to be the maternally provided mRNAs are now following here on. The maternally provided mRNAs are going to be slowly going down, and the levels will be down because they're going to get degraded. And as the maternal components are not completely down, because it's not that they're completely destroyed or degraded, they're substantially lower. The development is now taken over by the zygotic genome. And at this point of time, it is called maternal to zygotic transition. Now, so the question is, what are the events? So from stage 13 to 14 onwards, it is going to be the zygotic genome that has taken over. Now, it's not that the maternal components are going to be completely gone, because the maternal components are seen all the way to larval stages as well. Some of the maternal proteins and RNA are so stable, they survive all the way to late larval life. So many mutations which are lethal, they don't show lethality. So if you make a fly, if you're making a fly lacking notch, say for example, you may not find lethality. I mean, notch mutant flies are lethal, they don't survive, but some mutants survive all the way to larval stages. But post-larvae, they don't survive, because now how do they even make it all the way to larvae is because the maternally laid notch RNA is so stable, it can be translated and allow the early divisions or early development all the way to larvae happen. And now, since that is because of the maternal component. Now, if you remove the maternal component, then notch mutants, so those are called, if you make maternally null, so if the mom doesn't lay any notch RNA, then those embryos don't even survive early stages of embryogenesis, so they die. But if they are, if the genetics is such that the maternal components will be laid, and only the embryo is not going to be able to make a functional notch protein, then one can see the effects of maternal contribution. So it allows early development to happen, and sometimes all the way to late larval life, all the way to third instar larval life, the animal can survive. Okay, so now what is seen is that for the maternal components to slow down and the zygotic genome to get activated, it's not something that happens randomly. Now, it's quite a controlled event. While there may be many factors that control this maternal to zygotic transition, one of the few factors that control is the nuclear to cytoplasmic ratio. Now you can see that from the first cycle of nuclear division to stage 11, when there are around 256 nuclei, you can see that gradually the nuclear content or the chromatin content is going up, and the cytoplasm is going to be gradually divided into so many different nuclei, right? So the increasing nuclear content, and so the increasing the chromatin to cytoplasmic ratio, and the presence of transcription factors that are going to initiate the switch for the zygotic genome to get activated. Sorry, is there a question? Oh, does anybody have a question? No.
Question.
Hello? Can you guys hear me?
Yes.
Yeah. Yes, yes.
Okay. So the increasing amount of DNA, while the cytoplasm is actually constant, is one factor that drives the activation of the zygotic genome. Now, there have been experiments that, so how did this, so if you were to have haploid Drosophila embryos, so if there are half the number of, half the quantity of chromatin, so if you reduce the quantity of chromatin, then what was observed is that the divisions take much longer. And what is seen, what happens when an embryo is at cell cycle 8 for so if we have a haploid embryo, so what is going to be the amount of contact at stage 8 for the haploid is actually seen at stage 7, or what is going to be the chromatin content at stage 10 is going to be in a wild type, in the haploid embryo, is normally seen at stage 9 of diploid embryo. So you're just reducing it into half. The development slows down. It is not as fast, or it's not as fast or the same as seen in a diploid embryo, meaning everything with the half, with splitting, with reducing the amount of chromatin, you can slow down the development. The division slow, the cellularization slows down, blastoderm formation slows down. So all of the following events slow down, which means that the nuclear content or the chromatin content controls the progression of the subsequent stages of development. So the nuclear content matters. How the nuclear content dictates the development of the embryo. Had it been maternally driven, then one wouldn't have seen any difference. But in haploid, the experiment done by Bruce Edgar et al., what they show is that the nuclear content does matter and define the subsequent stages of development. Now, the second thing is, okay, the nuclear content, but the first thing is nuclear content, but the second thing is the activation of the nuclear content. Now there are some, so what the mom the maternal components that are laid into the embryo also has transcription factors which will initiate the activation of the zygotic genome. So, I mean, I'm if you read papers, but if you read Gilbert, it tells you about one of the transcription factors, which is called the Zelda transcription factor. It is maternally loaded, but actually it's a zinc finger binding transcription factor. And it actually binds to promoters present in the zygotic genome. And the presence of the Zelda transcription factor. So as the development proceeds, the maternal Zelda RNAs get translated, and the buildup of Zelda protein will lead to the activation of the zygotic genome. And all the enhancers, all the promoters in the zygotic genome that carry binding sites for this particular transcription factor will be first transcribed, will be the first ones to be transcribed. So this kind of sets up the activation of the transcription of the zygotic genome. Apart from that, there are also RNA binding proteins which will bind to the maternal RNAs and tag them for destruction. So while the nuclear amount, the chromatin amount builds up, the activation of the zygotic genome is also going to be initiated. The destruction of the maternal RNAs, all of these three major events together are going to lead to the maternal to zygotic genome transition. And once that happens, cellularization, as that happens, as that is initiated, it will lead to events that will lead to cellularization and then the gastrulation.
Tina, I had a doubt. This is Ron here.
Hi, Ron.
Yeah, so you mentioned that there were 3 events that caused the maternal to zygote activation.
Of the many, 3 which will essentially mean, the 3 that I'm trying to specify here mean that the nuclear content or the chromatin content matters.
Okay.
Third, the down regulation of the maternally loaded components matters. And third is, even as the maternal components are down regulated, there is a proportion, there's a population of maternally loaded RNAs, which are going to be transcription factors that are going to lead to the activation of the zygotic genome. So, you know, the zygotic genome is not transcriptionally active. It is inert. Now, transcription factors which will lead to the activation of the zygotic genome are also going to be, will also be needed to step in, but they're not going to come from the zygotic genome to begin with. So that is also laid by the maternal RNA. They come up, so the maternal RNA, not that, so one has to think about it, because the mom lays in everything. It lays in RNA, it lays in protein. But it's not that everything gets activated all the time and everything is being made all the time. There is series of translation and RNA binding proteins that will time what is being translated when and how stable are they. So like the bicoid mRNA, I mean, we'll come to it in the subsequent lecture. The initial events will be the bicoid, Oscar, nanos, mRNA, which are going to define the anterior, posterior. Those are the first few RNAs that are going to be translated, and their proteins are very important for the early events in embryogenesis. But later on, as cellularization and all of these events have to happen, or the zygotic genome has to kick in, following around that time point, probably these RNAs that are set in for being translated later on in embryogenesis, like the Zelda transcription factors, are going to kick in. So Zelda transcription factors cannot be translated from the beginning, right? They would screw up so many things. So however these RNAs are, they are laden, but they're only going to be translated a little bit later in development, and their translation is going to generate transcription. And as they're translated, these transcription factors will now drive the activation of the zygotic genome. So timing matters. Yeah, so go ahead with your question.
Oh, no, I just wanted to confirm whether it was whether there was just the buildup that caused the activation, or like you mentioned just now, there's a timing event also that matters.
No, so buildup is important. Buildup is important, but when does it start to build up is equally important. Because you can imagine these are transcription factors, right? Now, they will go and bind to promoters, correct? With a defined sequence. Now, the affinity of the transcription factor to the promoter will matter, correct? Now, ones that have the strongest affinity, even if there is very little amount of this transcription factor present, the sequences that have strongest affinity for this transcription factor will be the ones to be first transcribed, right? But gradually, as the transcription factor builds up in amount, then even the lower affinity binding sites will be transcribed. So you get my point?
Yeah, I think I do. So the buildup matters, but there is also some amount of
Timing.
Timing that also matters. Yeah.
Correct.
Okay. Yeah. Thank you.
Yeah. Okay, so I will probably want to show you, is my screen visible?
Yes, Tina.
Yeah.
So if I play a video, you'll be able to see it? So can you see the video that I'm playing right now?
No.
No.
No? Okay. What do I do to share?
I think you need to stop sharing the screen and then share the screen with the video.
Okay.
Yeah.
Okay. Okay.
This is a scanning EM. It's a
Wait, sorry. New share. Can you see this?
Yes.
Okay, great. So, okay. So this is now what I meant was the ventral furrow. So you can see that the cells are moving in, right? And as the cells are moving in, these are the ventralmost cells, and the mesoderm is going to get specified. And this indentation is the cephalic furrow. And that indentation there is going to give rise to the anterior midgut. And so these are the first rounds of morphogenetic movements that are going to be set in. And yeah, you can see now all the cells are moving in. The cells are moving in, and there's a movement from this end, which is going to be the germ band is extending. Now I'll show you one more video where it's perhaps more clear. I mean, you can see. Okay, now this is, yes, movement happens. The germ band is extending, and now it is going to retract, hopefully. Sorry. Wait, let me see this. There was one more video. Yes, this one. Okay, so as you can see here, the pole cells, this is the germ band extension. And now the segmentations are beginning and organogenesis is going to happen. The different structures are going to, the muscle, the gut, the fat body, all the different ventral nerve cord, everything is going to be initiated. Now this is the germ band retraction. And this is the amnioserosa, the cells of the yolk sac. And now what I meant about the dorsal closure. So now this is completely open. Now what is happening is this dorsal closure, you can see the cells are moving inward and the cells of the amnioserosa are being removed. I mean, actually it's led by cell death. Now these cells move, this is called dorsal closure. And now this is actually, this part what you see here is the ventral nerve cord. And now you'll see the embryo start sticking. This is sticking because the musculature is now active. The muscles have been formed in the earlier events and now the muscles are active. So there's a lot of movement that happens. And yeah. So I can show you one more time. The cells of the ventral furrow move in, germ band extension, cephalic furrow, the anterior gut moves in, the hind gut also moves, the gut from here, the posterior hind gut has moved in here, the pole cells have been placed inside, the segmentations are happening, and the germ band is now retracting, putting the yolk on top, and the cells move in to close from both sides, removing all the cells of the cells here. We're going to move in, removing the cells of the yolk sac. And you can see that there is a fine line. I'll show you a dorsal closure movie as well. And this is called dorsal closure. Following this, now the embryo has been completely patterned. The ventral nerve cord here, this is the ventral nerve cord here. And since the musculature is active, and at this point of time, there's still a lot of organogenesis is happening. All the larval structures are being defined. The imaginal discs, so the imaginal discs, which are structures that are going to give rise to the adult tissues, are also going to get specified in these later on stages of embryonic development. Okay, and this was the video that I wanted to show you about the early divisions. So you can see it's like a beautiful dance that the nuclei perform, very synchronized. And now they're going to move all the way to around 256 or around 256 nuclei is when they will start moving to the periphery. Yeah. Okay, wait. And this was an actin video that looks, so you can, so one point which I forgot to mention is that you would assume that the cytoplasm or whatever the cytoplasm that is there next to within surrounding each nuclei is going to be the same. It's not. The cytoplasmic determinants are not uniform across the embryo. It is not the same. And we'll come to that maybe in the next class. Yeah, and you can see that how the actin filaments are so synchronized. So these are the enegrids, these are the, I'm forgetting the name. Yeah, and the divisions are within a certain plane and looks very synchronized. Okay, I want to show you dorsal closure. Yep, this one. Can everybody see it, right? Yeah.
Yes, can.
So this is how the closure happens. So the cells of the amniocerosa are missing, but the two ends of the embryo, you can see how the, and yeah, and you can see how these cells are moving towards to stitch it. And there is this ring of actin, this cortical actin is very critical. It's like a very tight rope that seals it. And the movement of cells, I mean, now you can see the cells of the amniocerosa in this one. This is the actin ring that gets formed. And these cells of the amniocerosa are actually going to be deleted. But you see how actin structures are very important in, going to be very important in this closure of the two ends of the embryo, epithelial sheets. Okay, so maybe what will be good is if you guys can read upon, because this dorsal closure is an active, I mean, in Drosophila, this dorsal closure is not just for understanding embryonic development, but it becomes a fantastic model for cell migration, wound healing. So wound healing response, where the wound needs to be closed, people often utilize this dorsal closure as a ways to understand wound healing and wound closure response. Okay, what was the other thing that I wanted to show? Yeah, I think now that we have, let me stop share. Okay, so now that we have, I think I will stop now. So we have kind of covered the formation of the three germ layers. I mean, I'll come to the three germ layers more categorically next time. But essentially, the events of gastrulation are going to give rise to the structures of the larvae and also layout structures or cells that are going to be important for adult development. So now, yeah, so you guys have the slides, maybe you can go over it, and maybe next time on Wednesday, if there are questions that you have, we can discuss. Yeah, so these are, so now what I will start next class is how does the embryo get defined and determined, and how does the earlier, like I mentioned, the mRNAs, the maternally lated mRNAs and the proteins are going to be very important for the early rounds of cellular division. We'll pick it up from there on. And this is where Drosophila becomes exciting, because all the genetic screens that were done, the three essential screens that were done, led to the fundamental discoveries that now have become so, fundamental discoveries that kind of universal across different systems. So we'll start from there. And yeah, if there are any questions, I'm happy to take them.
Oh, okay.
Yeah, go ahead.
Oh, okay. I had a question.
Okay, Anandita, right?
Yeah.
Okay.
So you stated that the cytoplasmic determinants surrounding the nuclei when the syncytium is being formed is not uniform.
Mm.
So how do those cytoplasmic determinants, which are unique, are prevented from being diffused out and affect other cytoplasmic determinants which are from a different nuclei?
Yeah, so they're going to be RNA binding proteins, and how the So we'll come to that next class. So essentially, if you look at picoid or nanos, how they are laid out into the egg to begin with is not the same. So when the mom lays in the maternal components, it doesn't do a uniform job. So the egg itself has an anterior and a posterior polarity. So the cytoplasmic components are not the same to begin with from the anterior to the posterior end. And the way it does it is also by controlling RNA. You need RNA binding proteins that will be differentially laid across the embryo. And we'll touch upon that when we talk about this point too. As the egg is being constructed, the cytoplasmic determinants are not going to be the same. And we'll touch upon that in the next class. So essentially, the egg is not uniform from the anterior to the posterior to begin with. And the mom does it like that.
Okay.
Yeah.
Okay. Hi, I'm Meghna. I had a question.
Sure, go ahead, Meghna.
The RNA binding proteins that bind to the
Meghna has a
maternal.
question.
Sorry. I mean, yeah, there's a lot of background.
Hi.
Yeah, I can hear you now. Yeah, go ahead.
Okay. RNA binding protein that binds to the maternal mRNAs, which mediate its destruction. These proteins are maternal or
Everything is maternal. Yeah. So in the beginning, everything will be maternal, but once the zygotic genome kicks in, there will be RNA binding proteins that will even lead to the destruction of the maternal components.
Yes, but these RNA binding proteins are maternal or from the zygote?
So that's what I said. In the beginning, when the maternal RNAs that are being translated and present, or the maternal proteins that are present, they are going to be there for some time, and the maternally loaded RNA binding proteins are also there, which are going to time the destruction of the maternal proteins.
Okay.
Right? But gradually, as the zygotic genome gets active, it will also result in synthesis of RNA binding proteins, which will lead to a further destruction of the maternal components.
Okay.
Right? So once the zygotic genome is like on the driver's seat, it doesn't want the mom to be maternally loaded things to take over, because now the zygotic genome is going to do based on what it sends and how it's going to be coordinating the development now. So it is also going to be then any maternal RNA that hangs out much later in the larvae or in the later on in later larval stages is going to be degraded by the zygotically driven RNA binding proteins of the zygotically driven machinery.
Yes.
Yeah.
Okay. Okay.
But the initial switch from maternal to zygotic is driven by the maternal components. But these RNAs can be very stable. So that the maternal RNAs can be very stable and have very long poly A tails. And so that could make them very, very stable. And that is true, because even later on, not just in flies, but in even mouse embryos or any other developing eggs, if you see, embryos, if you see, these RNAs, the maternally loaded RNAs, can hang out until much later in even in Drosophila, they are stable all the way till late larval stages. Only in the pupal stages is when you start seeing them going down. So they are that stable. But they may not be the ones that are necessary for the zygotic transition. So the ones that have been the first to be degraded will be the ones that prevent zygotic genome activation. Because what the maternal components also ensure that you don't want to have the zygotic genome getting transcribed way early, right? So the first to be degraded are the ones that inhibit zygotic genome activation, like the small proteins. But thereafter, once the zygotic genome has been activated, then anything that is maternally loaded and will be also degraded by the zygotic component.
Okay, thank you.
Yeah. Okay, so more questions? Okay, great. So I have so one of the things that I want as the kind of what do you call food for thought is why do you think Drosophila development sees this kind of a syncytium based development? What are your thoughts? I don't think you'll have a answer to it, but it's the way you think about development. And as opposed to say mouse or Xenopus, where it is cellular divisions from the beginning, what could be, I mean, what are your thoughts in it? What could be the fundamental differences between cellular divisions that are seen in some developing embryos versus the syncytium based development that is seen in this Drosophila embryos? So that's one food for thought. Appreciate any thought, I mean, as long as you're thinking about the problem. Yeah, and then if you come across some better videos that show what we just saw today, yeah, feel free to share so we can go over them. I mean, if you send the link beforehand, then we can see it. Then I can share it. So if you have a video that will be a good thing, send it to me. Send the link to me, and I will share it next class with everybody else. Okay, great. So this was good. So I didn't speak to the screen. I spoke to people, and I'm glad about it. And I don't know how long the class is, but it is supposed to be 10:30, or we spoke for a long.
It's still 11 now.
Oh, great. Okay, fine. Good. Great. Thank you so much. And we'll see Wednesday. We'll meet Wednesday, 9:30. Okay.
Thank you.
Thank you. Bye-bye.
Thank you.
Bye.
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 Thank you.